Hi, and welcome to the 2.1
His specification, just in case you want to read
cells
through,
video for
pause it and
AQA
read if you need
A-level biology. So we're going to look at the optical microscopes versus electron microscopes. Now, optical microscopes can also be called light microscopes, and they're the microscopes that you would be used to using in classroom labs. You can see here a picture of your typical optical microscope. Optical microscopes are small. Obviously, you can fit lots of them in a lab. They are cheap, which means that schools can afford to buy lots of them so that all students can use them. They're easy to use, which means that you don't need any formal training in order to use them. You can have a couple of minutes of direction from a teacher at the beginning of a lesson and then be able to use them relatively successfully. And they give you color images. The limitations of optical microscopes are that the magnification and the resolution are both relatively low. So you can't magnify very, very small things like prokaryotes and viruses. And low resolution means that the focus isn't very good. So the ability to distinguish between 2 points is low. So it means that 2 points might look like the same point and be quite blurry. This here is an electron microscope. Now you can see the electron microscope is far larger and much more complex in its design. You typically wouldn't find these in a school lab. You would find them in university labs or research labs. They are super expensive. Another reason why you wouldn't find them in schools. And there's two different types. You've got transmission electron microscopes and scanning electron microscopes that we'll look at in a minute. Now, transmission electron microscopes give you 2D images, but they allow you to see inside the cells, so you can see the organelles from within the cells. And transmission electron microscopes have a really high resolution, so their focus is really good. Limitations are that only black and white images can be produced. So if you ever see an electron micrograph, which is a picture that we get from an electron microscope, if you see a micrograph with color, then that's been colored in after the picture's been taken. The image would have originally been in black and white. The specimens must be really thin in order to see them, and the specimens must be dead as well, because the specimen has to be placed within a vacuum. We've also got scanning electron microscopes. Both of these types of microscopes use electrons. They fire beams of electrons at your specimen, and that helps to visualize the specimen, whereas in an optical or a light microscope, you're just using light to visualize. Scanning electron microscopes produce 3D images, so it allows us to see the structure of a cell, like the whole structure of cells, or we could maybe look at things that are on the outside of the membrane. We could look at the shape of organelles rather than just looking at a cross-section of organelles that you would do with a transmission electron microscope. Scanning electron microscope has a slightly lower resolution than the transmission electron microscope, but a far higher resolution than the optical microscope. You see here that we have abbreviated transmission electron microscope to TEM. Scanning electron microscope can be abbreviated to SEM in exams as well. Black and white image is also produced from scanning electron microscopes, and specimens must be dead because, again, they have to be placed in a vacuum. They don't have to be thin, because obviously you can place whole cells or whole organisms into a scanning electron microscope so you can see its overall shape. Okay, on to calculating magnification. So we can see here the triangle for magnification, which I assume you've used previously in physics or maths. Now, magnification, we can see, is the image size. So that's the size of the image as measured with your ruler, divided by the actual size. Now, the actual size is the real life size, and we generally measure that in micrometers. You can also calculate the actual size if you have the image size, which again, you can measure that with your ruler, and if you're given the magnification. You won't ever be asked to calculate the image size because you can measure it with your ruler. Okay, so we also need to know that 1 millimeter is equal to 1,000 micrometers. So 1 millimeter here on your ruler can be further split up into 1,000 micrometers. Now, the reason that we do that is because obviously some cells, organisms are smaller, and organelles are smaller than 1 millimeter. So we need a unit of measurement for them. Micrometers is what we use. Now, let's take a question. So the actual diameter of this white blood cell here is 8 micrometers. So that's the actual size, 8 micrometers. They've given us that information. What is the magnification of the image? Now, the first thing that we do is we write out the equation. So magnification equals image over actual. If you cover up your M here, image over actual. So we write that out because we need to show all of our work in the calculation questions. Now we've filled in what we already know. So we know that magnification is the image size, we'll take care of that later, over the actual size. You've been given the actual size here, so that just goes straight in there. Now the image size here is 11,000 micrometers. Now we know that because we've used our ruler and we've measured from here to here. That's 11 millimeters. So 1.1 centimeters, 11 millimeters. That's the same as 11,000 micrometers. Remember, 1 millimeter, 1,000 micrometers. 11 millimeters, 11,000 micrometers. So we pop that here over our actual size of 8 micrometers. And we get a magnification of 1,375 times. So this image here has been magnified 1,375 times. Now we can also calculate actual size. So if our question was that the image has been magnified 2,500 times, what's the actual diameter of one of the red blood cells? Now, in an exam question, they would usually tell you exactly which red blood cell they will want you to measure, unless they're all a very similar size. So we start with actual size is image over magnification, so A equals I over M. Write it out in the question every time just so you don't forget it and you don't get it wrong. We've got actual size is image over 2,500. So they told us that the magnification was 2,500. We put what we already know in there so we don't get confused. The image size here, you can see the image size is 16 millimeters, 1.6 centimeters, 16 millimeters. Remember, 1 millimeter is 1,000 micrometers, so 16 millimeters is 16,000 micrometers, divided by our magnification of 2,500. And that should give us an answer of 6.4 micrometers. So the actual size, or the actual diameter of this red blood cell is 6.4 micrometers. Okay, on to cell fractionation. So cell fractionation helps to isolate specific organelles for further study or testing. So if you wanted to look at the mitochondria for further study, perhaps you're doing a project on mitochondria, you can use cell fractionation in order to isolate just the mitochondria. Now, the stages of cell fractionation, number 1, homogenization. What we do is we essentially put the cells into a blender, which breaks the cell membranes, and that releases the organelles into an ice-cold isotonic buffer solution. Now, it has to be ice cold in order to minimize enzyme activity, because obviously temperature affects enzyme activity. It has to be isotonic to stop any net movement of water into the organelles, because this would cause them to burst. And it must be a buffer solution to maintain a pH. So the purpose of a buffer solution is to maintain pH. The solution is then filtered. And when you filter the solution, it moves any large debris or whole cells that haven't been broken up in the homogenization stage. And then finally, ultracentrifugation occurs, and that means that your solution is spun at a really high speed. Now, when you spin the solution at a high speed, you end up with a pellet forming at the bottom. So these green circles here represent the pellet that's formed at the bottom. Now, above that pellet, we have a liquid. So this here is called the supernatant, and this is representing the liquid. What we do is we take that liquid out and put it into a new tube, and we spin it at a higher speed. So you can see here that this increasing speed of the centrifuge. So we spin it at a much higher speed, and then here we can see we've got another pellet. So then we take out the supernatant, we put it into another tube, we spin it at an even higher speed. So you get this pellet here into another tube, and then it's spun at an even higher speed. Now what's happening is these pellets are made up of the most dense organelles in the solution. So here you can see the green circles are representing the nuclei from the cells. Now the nuclei are the most dense part of the cells, so therefore when you spin at speed they kind of fly to the bottom and form this pellet. Once we've isolated the nuclei, the next one will isolate the mitochondria, and then the next most dense is the lysosomes, the next most dense would be the endoplasmic reticulum, and then finally this would happen again in order to isolate the ribosomes. If you are using cell fractionation with plant cells, the chloroplasts would be slightly more dense than the mitochondria, so they would come out second. You should know this order of isolation. They sometimes ask this in exam questions. So you need to know this. You need to know the reasons for the solution being ice cold, isotonic, and a buffer solution, and you need to know these stages. They're the most common exam questions.